I think this is the time to start our summer course today. Good morning for all participants from Europe and good afternoon for all participants who came from Asia and Asian countries. We are delightful to have you today in our third week's schedule of our summer course seminar. My name Muhammad Prima, and I will become your moderator for today's session. Okay, operator, can you next the slide? Yeah, this is me. My name is Muhammad Prima, and I will become the moderator for you today. I was graduated from the University of Tokyo, and my research interest in bioactive peptide, biotechnology, and fish processing. So I'm welcome if you have an interesting in this kind of research scope, then we can make a collaboration. And today we already have three very outstanding researchers in the field of fish product technology. I will introduce you one by one, our speakers today. We will start from Dr. Lotta Kuuliala. Good morning, Dr. Lotta. Yeah, we meet again.
Good morning.
We have ever met in our international seminar, maybe two years ago or last year's?
Last year, yes.
Last year's, yeah. Oh yeah, last year's, yeah. Thank you very much. Yeah, you still, welcome to our department. Thank you for coming today. So Dr. Lotta is Junior Postdoctoral Fellow of the Research Foundation Flanders at the Research Unit Food Microbiology and Food Preservation and the Research Unit Knowledge-Based System at Ghent University, Ghent, Belgium. Welcome to our summer course, Dr. Lotta. And Dr. Lotta's activity is focused on data-driven method within the context of intelligent packaging technology development, with a particular focus on volatile-based seafood quality monitoring. Very, very interesting topic. Thank you, Dr. Lotta. The next speaker will be Dr. Chitsiri Rachtanapun. Good afternoon, Dr. Chitsiri.
Good afternoon.
Am I right on spelling your name?
Yeah, I'm Chitsiri, yes.
Chitsiri. So welcome to our department. Thank you very much for coming today. So Dr. Chitsiri is an assistant professor at the Department of Food Science and Technology, Faculty of Agro-Industry, Kasetsart University, Thailand. Yeah, our neighbor. Thank you very much for coming today.
Very close.
Dr. Chitsiri expertise is in food microbiology and safety of natural antimicrobials. Thank you very much for coming today, Dr. Chitsiri. And our next speaker is, I think the first slide. Moderator, can you next slide? Yeah, so our next speaker is Dr. Indun Dewi Puspita. Good afternoon, Dr. Indun.
Good afternoon, Mas Prima.
Yeah, thank you very much for becoming our speakers today. So Dr. Indun is a lecturer and researcher in the Department of Fisheries, Faculty of Agriculture, Universitas Gadjah Mada. So he got his doctorate from Hokkaido University, Japan. And her research interest is in applied microbiology and fish processing. Thank you, Dr. Indun. Okay, so thank you again for all three speakers for this session, and all is women. We are delightful to have three women speakers today. Thank you very much. And also welcome to all participants. Yeah, we still have 28 participants, -3 and -4, so 24 participants, excluding our committee. Yeah, we still have from Myanmar. Good morning, good afternoon. Ninkat. We have Hatat Wailin from Myanmar. Good afternoon. Welcome again, Kelvin Ong Zhao Kang from Malaysia. And I can see another participant from another country. Yeah, only from Myanmar and Malaysia. And another participant is from Indonesia. Oh, there's also Susana Enda. Good afternoon. Oh no, good morning, Susana Enda. Nice to have you here. Okay, so without delay, we will start our session from Dr. Indun, right? Dr. Indun, are you ready?
Okay, I'm ready.
Okay, time is
Mas
yours.
Prima.
Thank you very much.
Okay. Thank you very much, Mas Prima, for a very nice introduction about me and also my old friends. I'm really happy that I can see Dr. Chitsiri and also Dr. Lotta again. For Dr. Chitsiri, I have no contact and also I haven't seen her for quite a long time, and I'm so happy that I can see her again today. And Dr. Lotta, thank you very much. You're accepting our invitation again. So, Mas Prima, may I share my own screen?
Yeah, please.
Okay, good afternoon and also good morning for all of the participants. So today within 30 minutes, I hope I can finish all my lecture today. I will share the topic about the fish handling practice and also the characterization of histamine producing bacteria. And this is the case study from our research group, which located in Sadeng fishing port in Yogyakarta. But before I go in detail in the result of our case study, let me start my presentation with this map or with this graph to show you that globally, tuna, which is signed by the graph with the blue color, is the main fisheries capture in the world. And if we see more detail in the Southeast Asia, this one probably is in Indonesia, the area of 71. Tuna is one of the biggest capture production from Indonesia. So this is one of the important commodities in Indonesia, and Indonesia is one of the big producers of tuna in the world. And as we know that tuna is really delicious, I think that many people like tuna, one of the fish from the marine. And we also know that tuna has a very high nutrition, including protein and also mineral and also vitamin. And except of this good nutrition value of tuna also has the complete amino acid such as histidine, arginine, threonine, and so on. But one of the concern with this tuna within this nutrition value is the content of histidine. So compare with other seafood, tuna and their friends is high in histidine. So this is the potency, or it has a higher potency in contaminated by histamine because of this content histidine in tuna. And histamine is one of the safety standard for tuna product, and it is globally accepted in the tuna trade. However, there is still some problem in the regarding to the safety of global tuna trade. And histamine is one of the concern in this food safety regarding to tuna product. Many of the reports we can see in the news globally or also regionally, and this is I tried to find in the internet. In 2019, histamine is one of the causing of outbreaks in Europe, and it is predicted to be increased because Europe increasing their import of tuna. And also in Canada, some report said that tuna, which comes from Asia have to be recalled due to the high levels of histamine. And if we see the newer news, like in 2021, histamine is still the problem in the trade of tuna. In Sweden, they report the histamine outbreak, which is come from the fish which is imported from Vietnam. And latest in 2022, I found the news, which is one of the supermarket in Spain, they have to recall their frozen pizza, which is written as the frozen tuna pizza because of the high histamine detected in these products. So that is, I would like to show that histamine is still a concern of the food safety for the tuna product. And in Indonesia, we also face the same problem about the histamine from the fisheries product. This graph, I would like to show that in 2010, during 2010 until 2020, this is the study of our student in department, Nur Hasana et al. 2022 reported the amount of the rejection cases from the Indonesian fisheries product in the USA. So we can see from this graph, tuna is the biggest product which is reported to be rejected in the United States market, followed by shrimp and also snapper and so on. But from this graph, we can see that even tuna is one of the biggest export commodities from Indonesia, but it also still has the problem due to the safety of this product. And this problem seems to be happen every year, because this is 2010 until 2020, and the report said that every year the rejection of tuna is still happen. So I think there is still some problem which is have to be taken over by many person or many party, which is including or related to the global trade of tuna. And if we see more detail to the rejection causes of Indonesian fisheries product, this is the Pareto diagram. This is still from the same study. We can see that filthy and salmonella is the main reason of the rejection. But we can also see histamine is in the 5 causing problem of the rejection. So I think this histamine is still homework for many of the industries and also government to ensure the safety of the tuna regarding to the histamine content. And in Indonesia, actually, we have the standard we call the Indonesian national standard regarding the histamine concentration of the seafood product. And this is type of the product you can see from the fresh one to the frozen one. And mainly it is based on the product is the tuna-based product. And the concentration is depend on the product, which is range from 50 to 100 milligram per kilogram of product. So when the Indonesian industry would like to export their product, mainly for the tuna product, they have to meet this standard. It means that all the histamine content of the product should be under this value. However, it is still a big question why, I mean, all the product which is export is have to be meeting with this standard. But after the product reach to the countries, the other countries, which is the like the USA that we saw previously, why the rejection is still a homework and also a big question. Whether the histamine is caused or it's increased during the transportation, or probably the standard is different, or probably the method to analyze is could be different. So I think this is have to be a concern for Indonesian fisheries. And if we see domestically, or in the Indonesian itself, histamine is still one of the main cause of food poisoning outbreak in Indonesia. I'm sorry, this is still in Indonesian, but I can show you, or I can tell you that this graph shows the number of cases of food poisoning outbreak in Indonesia, which is caused by microbiology and also by the chemical compounds. If we see in the chemical compounds, there is still 12.6% of the causing agent of the food poisoning outbreak. And inside of this value, this is histamine, which is the cause of food poisoning outbreak. And also this histamine poisoning outbreak is happen every year. Is it reported every year? So I think regionally in Indonesia, histamine is also still a concern for the food safety. This data I got from the surveillance report from the Yogyakarta Fish Quarantine Agency. They try to survey the histamine content of the fish, which is sold in the traditional and also modern market in Yogyakarta, so in the local Yogyakarta. And it turns they found that 64% of fish which is sampled from this market, so 14 markets in total, 64% of the fish sample contain histamine higher than the standard. So it must be something happen so that the fish which is sold in the market contaminated by this histamine. So actually, why histamine is become a concern in the food safety? Because histamine can someone, when they consume histamine, they can be poison, toxic, which the nausea, vomit, diarrhea, and also oral burning. Actually, the symptom is not very harsh. I mean, there is no report that histamine can make someone dead, but this kind of symptoms, some people can have the more severe effect compared to other people. So it depends on their immunity. And how the histamine can be produced or can be contaminated of fish, it is start from when the fish is dead. So when the fish dead, all the oxygen supplies is ceased or stopped. Then the reaction of anaerobe make the pH of the fish reduce because the accumulation of the lactic acid. And this acid, the acid environment induce the decaying of the fish further. As we know, or all that, or many people knows that this low pH can induce the autolysin, which is protease, which can degrade protein in fish more further into the small peptides, which the small peptides, or the I say a smaller or the simple protein, can be used by the bacteria, especially for the decaying bacteria, to proliferate or to grow. Then the fish become decayed. But in other side, beside of this protein degradation, some of bacteria can use this condition to grow better. And one of this group of bacteria is histamine producing bacteria, because they can produce, or they can produce enzyme, and also make the environment which is low and it is not optimal for they grow into the higher pH, which can make them optimum to grow. So this is the illustration. So when the environment of the living environment of the bacteria became to decrease in pH, then they try to find a way to survive. Some of the bacteria, or probably the normal bacteria, they cannot live in the acidic environment. So this bacteria try to survive by changing histidine, the free histidine in the fish protein, to become the histamine by producing histidine decarboxylase. When histamine is accumulated in the environment, then this histamine can make the pH higher so that the bacteria can survive and grow better. So some of the reports said that with the lower acid, histamine producing bacteria is induced and produce histamine higher than in the normal environment. So that's part. I would like to show you the important or the significance of histamine in the global trade of tuna, and also concerning the food safety of the tuna. And then we move to the what we have done in our research that we would like to share all of you is our study in the Sadeng fishing port. So Sadeng fishing port is one of the big port in Indonesia, especially in Yogyakarta. And many of the fish which is distributed in Yogyakarta is come from this fishing port. And we try to find what happened in the handling of the fish in this port by measuring the time of handling, and also by measuring the temperature of fish during the handling step. So we try to measure the fish in the ship when the fish is put out from the ship, and also when in the fish auction. So after the fish was collected, and then they will be sold to the seller in the this location, the fish auction. Actually, the location is inside the fishing port. And also after the finish of the auction, and then the fish is stored in the warehouse, which they use the ice in this step. So from the ship until the warehouse, there is no cold chain in this step. So we try to measure the temperature of fish. We try to measure from the two type of ship. So the first one is the bigger capacity of 41 to 45 gross ton, which they can capture fish from 1 to 16 ton, and they have the time for fish tripping 6 to 7 days. And we found that the fish inside the ship, the temperature is slightly increased. So it's 8 to 16 degrees Celsius, which this temperature is, I think it's not good for the cold chain. And they handled the fish from this ship until the warehouse within 68 to 200 minutes. So it's quite a long time, and it makes the temperature of fish increase into 13 until 34 degrees Celsius. You can imagine 34 degrees Celsius is the ambient temperature. So after the fish reach the warehouse, then they put the ice on it. So I think something is happened between this handling step. And we try in the smaller ship, which is the capacity is 15 gross ton, and it can capture 2 until 7 ton of fish with 4 to 5 days trip. Within this smaller ship, they can maintain temperature better, because the temperature of fish is around 3 to 4 degrees Celsius, and they can handle fish faster within 100 minute. But however, the temperature of fish is still increased. We found that the temperature become 15 to 22 degrees Celsius. So I think this handling might be one, or probably is the one factor that makes the histamine produced during this temperature abuse. And we try to identify what is the histamine producing bacteria that may be found from the fish landed in this fishing port. And we found 7 genera of bacteria, not 7, I'm sorry, 8 genera of bacteria, Rotella, Citrobacter, and so on. And the dominant genera of bacteria is from Pseudomonas, and the second one is the Morganella. And all of these bacteria is grouped as the Enterobacteriaceae, which is the main producer of histamine. And we try to characterize some of these bacteria in terms of growth temperature and also the addition of salt, because usually salt is the simple preservation method which is practiced by the fishermen and also the small-scale producers in Yogyakarta. We try to find out how temperature can affect the growth of histamine-producing bacteria. We try with these 6 isolates, isolate from the skipjack tuna. This is the species reference, which is we identified using the molecular method, using 16 srRNA. And we found that in 5 degrees Celsius, actually, all the isolates are inhibit to grow. But within 20 and 37, the growth rate of the bacteria is varied. We can see from this 6 bacteria, CK4, which is identified as Morganella, has the higher growth rate. When we see the production of histamine during 20 and 37 degrees Celsius, not the CK4, which is produce the highest histamine. Instead, CK6 is the one who produces histamine higher. So from this finding, we can see that not all the bacteria which grow better in this temperature will produce the highest histamine in the same temperature. And also we try to find out about the addition of salt to the growth rate and also the histamine production. We try to use 3 isolates, Klebsiella, Morganella, and Rotella, which is these 3 isolates produce histamine highest compared to other isolates. And we found out that the CK2, the blue one, the Klebsiella, they can produce histamine better in the presence of salt compared to the other isolate. And also, the more interesting is that this isolate can produce histamine earlier than the other isolate. And we found, or we hypothesized, that this histamine-producing bacteria can contaminate other fish using the method of biofilm. Can produce biofilm, and when the biofilm is matured and dispersed, the active cell or the planktonic cell can contaminate to other fish. And this biofilm can be produced in the surface of the machine or the surface of the container which is used in the handling of fish. So we try to find the ability of this histamine-producing bacteria in forming biofilm. We use the crystal violet method after we make the the bacteria produce biofilm, and the biofilm is measured by the crystal violet, which is washed from the container. And we found that the some of the histamine producing bacteria can produce biofilm in the weak biofilm, but some of them, or most of them, can produce a strong biofilm. strong biofilms make these bacteria can survive longer or stronger in the surface of the container. So it means that the potency of contamination can be higher. And also we try to make the simulation, because we we believe that in the biofilm, bacteria is not a single strain, but the mix of strain. And we found that the mixed strain of the histamine producing bacteria can make biofilm stronger than the single strain. So from this finding, we can propose the controlling method of histamine producing bacteria growth in seafood by three factors. First is to inhibit the growth of bacteria. We can use like the temperature and also addition of salt, pH, or other processing method to make sure or to inhibit the growth of bacteria. And we can also use another method like the bacteria competitors, for example, in the fermentation product, or the secondary metabolite from other microorganism was reported can inhibit the growth of histamine producing bacteria. And the other thing that we can do to control this histamine producing bacteria is to control or to inactivate the enzyme. Actually, I'm not review much about this enzyme, but the report said that because the histidine decarboxylation enzyme is inside of the bacteria, they are not produced extracellular. So when we inhibit the growth, or when we try to disturb the cells, the enzyme probably can be inactivated. So we can use also temperature, pH, or processing method. And some report they use the histamine degrading enzyme to reduce or to degrade this enzyme. And the other thing is we can eliminate the biofilm in the surface of the container by using the sanitizers or other chemical compounds to make sure or to inhibit the cross-contamination of these bacteria from one product to another products. So the take-home message from our sharing is that histamine is concerned as one of the global problem regarding to seafood safety, and we can find that various species from Enterobacteriaceae, which is common as the histamine producers found in seafood. And as we see, the characterization, which is vary between one bacteria to another bacteria in producing histamine, Hardel technology or the combine of the technology must be applied to make sure that the bacteria can be inhibited to grow and produce histamine in the product. I think that's all, Mas Prima, my sharing. Thank you very much. I give the time back to you.
Thank you. Thank you very much, Dr. Indun, for a very interesting talk about histamine. For those who have a question or interest in histamine and histamine producing bacteria, please keep your question and you can ask by the end of the third speaker speaking. Or you can also raise hand in the question and answer session. So we will continue our talk about fish product technology by welcoming Dr. Lotta. Dr. Lotta, are you ready? You have the same 30 minutes to talk, and then the question and answer will be in the last session. Please, okay. Dr. Lotta.
Thank you very much for the introduction and for the kind invitation to have this opportunity to present. I will try to share my screen. Let's see how I manage to do that. Okay. And yes, I hope you can see my slides.
Yeah, very clear.
Yes, perfect. Thank you. So indeed, I'm very glad to be here to discuss a topic called spoilage indicators for seafood quality monitoring. And this is a very interesting topic, but also a very wide topic because, well, seafood spoilage can happen because of many reasons, and it is affected also by many factors. And this also means that spoilage can often be detected in many different ways. So let's first have a little look at the outline of the lecture. First of all, I will start with a short introduction to seafood quality and spoilage indicators. So what they are, where they come from, what kind of factors affect them, and how spoilage indicators can be useful when thinking of quality monitoring. And after that, we will have a little bit more detailed look into some examples of studies where VOCs, so volatile organic compounds, have been used for seafood quality monitoring. The use of VOCs as spoilage indicators is something that has gained a lot of interest, and we will have a look what these are and why they are so useful, and what kind of things we need to consider when planning and performing experiments, and what kind of methods we can use for measuring them. And last but not least, there will be some take-home messages and future considerations in the conclusions. But first of all, let's start with a short introduction to seafood quality. Seafood in general, it's a very appreciated food product type, and it has great economic and ecological relevance. And there are, of course, many contributors involved in the processing and the distribution and retail, so processing industry, but also like regulatory, legislative authorities, and of course, consumers. And all these contributors have their own needs and expectations when it comes to seafood quality. But still, at the same time, seafood is generally known as one of the most perishable food products that there are. So for this reason, there's a great demand for rapid and objective methods that we can use for quality monitoring purposes. But in order to achieve efficient quality monitoring, we have to be aware of those many factors which are affecting seafood quality. And indeed, there are many factors to be considered, but these can be divided in three main groups. First of all, the intrinsic factors, which are related to the properties of the fish. Secondly, the extrinsic factors, which are related to the environment of the fish. And also implicit factors, which are related to the interactions of the microbes which are present in the food. So here I will focus on the first two types, so intrinsic and extrinsic factors. And when thinking of the intrinsic factors first, so the properties of the fish, there the chemical composition is of course a major factor. And there's a lot of variation when it comes to chemical composition. It can vary between different types of fish, but also, for example, according to the season and to the living habitats of the fish. Microbiota, on the other hand, is another major factor which has critical effect on how the spoilage processes. And indeed, seafood, it's highly perishable, and microbial activity generally in fresh fish is the most, it's the main contributor when it comes to spoilage of fresh fish products. But composition and microbiota are still not all, because all kinds of properties like the size of the fish, the pH, and the presence of natural antimicrobial compounds are all factors which can have an impact on the progression of spoilage. Then if we think of extrinsic factors, then there so something related to, for example, the living habitat of the fish, or for example, the processing environment. their temperature is the most critical factor, because temperature will largely affect how spoilage processes, and for this reason fresh products should be kept low. But also atmosphere, so the gases which are surrounding the product will have a major impact. And the technology of modified atmosphere packaging is indeed based on the idea of changing the gases which are surrounding the product, and so to extend the shelf life. I will indeed later, when coming to the examples, show some results which show how great the effect of the atmosphere indeed can be. So when thinking of quality as a concept, quality is a complex concept and it involves many different factors. So what we often speak about is like microbiological quality, sensory quality, so referring to the sensory properties like the smell and the taste and the texture of the fish, nutritional quality, and so on. And the different properties of the fish contribute to the quality status. But the quality status is of course not fixed, but it will change as a function of time because of microbial activity on one hand, chemical reactions like lipid oxidation, and physical damage. And these kind of processes will cause changes in the product. So for example, the emergence of odors, of flavors, and alterations in the texture. So generally we can define food spoilage as a process which leads to the fish product, or well, food in general, becoming undesirable or unacceptable for human consumption. So then if we consider how we can measure this, so how we can analyze and how we can measure spoilage, there are of course numerous methods how to analyze the quality of fish, and I won't go into detail into all of this. This list here is also by no means exhaustive, but it gives a bit of an idea of what kind of methods there are that are frequently used for quality monitoring purposes in seafood products. So for chemical methods, for example, biogenic amines and K-value, they are frequently used. But it should be noted, of course, that not all methods are applicable in all kinds of situations. So, for example, electrical measurements, which are targeting the properties of the fish skin, are applicable for whole fish, but not then for fish fillets, which don't have the skin anymore. And on the other hand, the methods are, of course, also used for different purposes. So when thinking, for example, of sensory evaluation, which is based on the use of human senses in the evaluation. So people performing, for example, smelling or tasting tests. There are also many different types of tests which can be used in different situations, like to assess consumer acceptance of a product, or to, let's say, evaluate if the panelists are able to distinguish an odor or if they are not. Then, in this scope, spoilage indicators, they can be defined as variables which we can use for quality monitoring purposes. So monitoring the quality and the spoilage of the food products. And as an example, we can consider the production of microbial metabolites, because microbial growth and metabolism leads to the degradation of chemical components in the food. So there will be metabolites produced, and this will lead into changes in the product, such as, for example, the emergence of odors and of flavors. And as time goes on, these changes become more and more prominent, and eventually, for example, the off odor becomes so strong, the product will be rejected as spoiled. So this means that if we are able to detect those metabolites and to measure the levels, we can use this for monitoring the quality of the food product, and so to distinguish fresh products from spoiled products. So here we are using metabolites as spoilage indicators. So it's a very interesting approach in many ways, but it's very important also to note that there is no single spoilage indicator that we can use in all kinds of situations, like for all kinds of seafood products and in all kinds of storage conditions. Because indeed all products have their own spoilage processes, which are affected by many factors. So this brings to the question how we can then know what are the useful spoilage indicators. So what should we be measuring then? Here we have to consider three very important aspects, which are the applicability, the feasibility, and the performance of the potential spoilage indicators. So the applicability means if a spoilage indicator can be used or not. So for example, not all metabolites are produced in all products, at least in high enough levels. And on the other hand, feasibility refers to, let's say, how easy it is to use a certain or to monitor a certain spoilage indicator. Because for some metabolites, it can happen that they have a very good correlation with microbial and sensory deterioration, but the concentrations are so low that we need really sensitive instruments to be able to detect them. And methodology in general can have all kinds of other limitations as well. So when thinking of microbiological analysis, for example, these are typically destructive, which means that we lose the fish sample that we analyze. So I will actually come back to these considerations a little bit later, because we are now moving to the second part, which are the examples related to the analysis of volatile organic compounds. So yes, I already spoke a little bit about metabolites. So continuing from there, volatile organic compounds are that kind of compounds which are basically responsible for the bad smell when seafood is getting spoiled. So the mechanism is indeed what I already mentioned. So when the chemical components of food are degraded, for example, because of microbial activity, but can also be other phenomena like, say, chemical reactions, lipid oxidation, there will be VOCs produced as a result, and this will lead to off odors. Not all VOCs can be smelled, especially if they are not present at high enough concentrations, but many of them anyway can be smelled. And this eventually, when the off odors become stronger and stronger, will lead to consumer rejection. So this means that indeed, by measuring the concentrations of VOCs, we can then monitor the quality status of the food product. And while this is something that has been possible in laboratory scale already for quite a long time, it's a more, let's say, it has more recently become a topic to use this as a basis of normal packaging technologies. And as an example of one of such projects, I wish to show this figure. This is a figure which summarizes the project Checkback. Checkback was a project which was coordinated by Ghent University a few years ago, and the idea was to develop a sensor. So an optical sensor, which can be integrated inside food packages to measure the concentrations of VOCs which are formed during storage time, and so to monitor the spoilage of the packed products. And this would indeed happen without the need to open the package, because the sensor is inside the packages. And eventually, this kind of approaches could be used for replacing traditional shelf life labeling, because the challenge is that when we print, for example, a use-by date on top of the packaging, this cannot take into account if, for example, the cold chain has accidentally broken down at some point during the distribution chain, and the actual shelf life no longer corresponds with the printed date. So with a sensor, we could get instead real-time information regarding the quality status of the food. So there's many advantages indeed, but on the other hand, the development of this kind of sensor containing packages is a complex task. And in order to be able to do this, there's a lot of information needed. Because indeed, all food products have their own spoilage processes, which are affected by many factors. So this means that it generally takes a lot of experimental effort to understand the spoilage mechanisms. And this means that when thinking of VOCs as spoilage indicators, we have to be able to answer two very important questions to be able to use them as a basis of novel packaging technologies. And these questions are, first of all, which VOCs are important? So which VOCs should we be monitoring? And secondly, how much is too much? So what are the critical concentration levels that we have to be able to detect and to measure, and so to be able to distinguish when the products are spoiled? So in the examples that follow, we can have a look how these kind of questions have been answered in the past in one study related to salmon. But just a little bit background information maybe before going into there, because there has been lots of interesting studies regarding VOCs in the past, and many different kinds of compounds have been identified in many situations. So typically, alcohols, aldehydes, ketones, acids, sulfuric compounds, amine compounds, there have been many VOCs already which have been found relevant in different kinds of products. For example, among sulfuric compounds and amine compounds, there are many such compounds which can be produced by many microbes. And these compounds are also often smelled at very low concentrations by people, and they have an offensive smell. So they contribute to the offensiveness that occurs when spoilage proceeds. So the examples that I will show come from a study that was done to examine the spoilage of salmon. So in this study, we packed salmon fillets under different types of modified atmospheres. There was atmospheres where no oxygen was present, so anaerobic atmospheres, and also aerobic atmospheres, including oxygen. And the samples were stored at 4 degrees until spoilage occurred. And at regular intervals during storage time, we took samples to be analyzed with microbiological, chemical, and sensory evaluation methods. And this picture here is showing the principle how the chemical measurements for the VOCs were done. So this is so-called selected ion flow tube mass spectrometry, where the principle was that we were able to extract gas samples out of the package and analyze the concentrations in real time. So coming then to the results. So in this figure, you can see a comparison between the VOC production in the different atmospheres. On the left side, there are the concentrations of day one, which is 1 day after packing. In this situation, there is not yet much VOC production, and there's not really a difference between the two conditions. But after 13 days of storage, the situation is very different. It can be seen that the concentrations have first of all greatly increased, but that there's also a major difference between the two different atmospheres. So under anaerobic atmospheres, especially ethanol and hydrogen sulfide have become much higher when compared to the aerobic conditions. So these results illustrate how big the impact of the atmosphere can be. And also that's very difficult to find like only one single spoilage indicator that could be used for all kinds of situations. It's actually not possible to only have one. But good news anyways are that some VOCs are anyway more common than others. So they have been found in many kinds of situations and many kinds of products. And as an example, alcohols are compounds which are frequently produced in many storage conditions and products. And for example, ethanol has been frequently detected in the past. So this figure is showing results from the same study as the previous one, but now only the quantified alcohols. And what can be seen is that when microbial growth proceeds, the VOC levels, they are initially low, but they rapidly start increasing when we go towards higher levels. And it has indeed often been seen that we need approximately 7 to 7.5 or 8 log counts before observing rapid increase in VOC production. So these results illustrate a little bit what kind of VOCs we found relevant. But then how about the second question? So how much is too much? We can have a look at third figure. So this figure again showing results from the same study, but now this is shown in relation to the sensory evaluation results. So on the x-axis there is storage time. On the y-axis are the concentrations of the VOCs. And if you have a look at the symbols, the closed symbol is indicating those concentrations which were still acceptable to the panelists who were smelling the samples. And the open symbols are then those concentrations which the panelists rejected as spoiled. So what can be seen here is that in the beginning, again, concentrations are low and the panelists are accepting the samples. But then approximately at day 5, rejection first occurs when concentrations are increasing. And at this point, interestingly, only ethanol is exceeding the range of 1 ppm, so parts per million. And this is indeed something which has been often seen, that we need to have ppm range concentrations, or let's say these concentrations are linked with the spoiled samples. But when thinking of sensor sufficiency, it's very important that we would not only be able to detect that kind of levels, which are indeed linked with clearly spoiled samples. So we must be able to detect like clearly spoiled fish, but we must also be able to detect earlier when spoilage is starting. So let's say early stages of spoilage. So this means that low sensitivity, or let's say we need to be able to detect concentrations at very high sensitivity in order to be able to detect also early spoilage in many cases. So as a final part of the lecture, I wish to say a couple of words about methods that can be used for VOC analysis. And there are indeed many methods, so I won't be going into detail with these, and not technical detail as well. But I wish to highlight some methods which have been frequently used in the recent past. So GCMS is one of these. GCMS is very versatile, and it combines two very efficient strategies. So gas chromatography, first of all, for the separation, and mass spectrometry for the detection of the VOCs. So there are indeed many adaptations and many applications for this technology, but one drawback is that the analyses are often relatively time-consuming and labor-intensive. So it takes time and effort often to achieve the results, especially if we need quantitative data. So one alternative is real-time mass spectrometry. So I already mentioned CITLAS, but for example, PTRMS is another option. And here the principle is that when the gas is traveling through the instrument, the VOCs will undergo chemical ionization and will be finally identified by mass spectrometry. So this happens.
5 minutes left,
Okay, I think I will manage. So this happens normally in real time, so that they can at the same time measure and on the other hand see the concentrations on a graph. But yeah, so many advantages. It's fast and real time, and we can measure exactly at commercially relevant storage conditions. But it should be noted that this is still an invasive method. So we still need to pierce the package in order to be able to take a sample from the headspace. So if we consider that kind of scenarios where the package can remain closed and intact, This could be achieved with indicators of sensors, which are both falling under the scope of intelligent packaging. So in this lecture, I have been talking about indicators as indicator variables, but here indicator means a little bit different thing. It's an indicator device. So for example, a tag or a label which changes color when it comes to contact with the compound produced. So this kind of tags normally provide non-quantitative or semi-quantitative data, whereas sensors, on the other hand, often provide or generally provide quantitative data. So on the whole, both technologies have many advantages: fast measurements, real-time measurements, but one common drawback is still the specificity in many cases, because typically these solutions can detect one VOC or a couple of VOCs, or let's say VOC groups, but mass spectrometry, on the other hand, can detect considerably more. So I think I will come to the conclusions now. So to emphasize that since there are many factors which are affecting spoilage processes, we need a lot of data in order to identify and quantify spoilage indicators in different scenarios. So when we are aiming at developing, for example, novel packaging technologies based on the VOCs, let's say, multidisciplinary collaboration between different fields is very important. And in the future, there is a lot of work still to be done to both extend and to deepen the current knowledge that we currently have, and to develop novel methods for quality monitoring. So with these conclusions, I hope that you have enjoyed listening to the lecture, and I wish to thank you very much for your attention. Thank you.
Thank you very much, Dr. Lotta. This perfect 30 minutes. So, we have increasing of the participants. Now we have 46. Thank you very much for all participants who already joined today. We will continue to our third women researcher today. So Dr. Chitsiri, are you ready?
Hello.
Okay, thank you very much.
Do you hear me?
Yeah,
okay.
Your time is yours. Yeah.
Okay. May I share screen? Okay. Do you see my screen?
Not yet.
Not yet.
Okay, it's loading now. Okay, waiting.
What's wrong?
So still like.
Yeah.
Okay, it appears perfect. Yeah, you have 30 minutes, Dr. Chitsiri. Time is yours.
No.
The slide.
Good afternoon, everyone.
Yeah.
First of all, I would like to thank the University of Gadjah Mada for inviting me to join this summer course program and letting me share my experience through the topic of Chitosan-based active packaging to promote seafood safety. And my name is Chitsiri Rachtanapun. I'm a lecturer at the Department of Food Science and Technology, Faculty of Agroindustry, Kasetsart University. And my specialty is food microbiology and food safety, focusing on natural and alternative antimicrobial substances. I will divide my topic into 3 parts, starting with the seafood safety concern and moving to the chitosan characteristic and its functional property, and then focusing on the application of chitosan as active packaging for seafood safety. Seafood is normally consumed with a minimal process, so we don't like the too cooked the seafood. So the hazards of the seafood mostly are chemical and biological hazards. These are the main issues of the food safety concern. These are the list of chemical and biological hazards related to seafood safety as described by Camden BRI guideline number 68. It's a comprising of Salmonella, Vibrio palahemolyticus, and Vibrio cholerae, Listeria monocytogenes, Clostridium botulinum. If the seafood was packed in the modified atmosphere packaging or packed in the vacuum packaging, and the toxin, the histamine and planktonic toxin are the chemical hazards that's mostly concerned in the seafood as well. And virus is another biological hazard, but mostly virus is transmitted by mostly a human. So when we talk about the foodborne pathogens, these bacterial poisoning organisms are more applicable to ready-to-eat products. But for the chemical and toxins, it's mostly related to cooked fish. These pathogens contaminate the seafood products where raw material, where the contaminated environmental undercooking process, or it may be the cross contamination during processing, the post-process contaminate. These issues can be compromised by the good manufacturing practice and other preventive control. But yes, such as the HACCP or using a preservative is one of the control methods to increase the food safety and extend the shelf life of the seafood product. But however, recent findings on consumer trends show that more and more consumers are looking for food products without artificial food additives, which are often regarded as unhealthy. But as these, so the clean label trend is gaining popularity, and it had led to the increased interest of natural and green ingredients, which would be accepted by the consumer, while exhibiting a number of bioactive properties. And chitosan is a polysaccharide biopolymer. It is a normal obtained from seafood waste, but actually it can get it from fungi and insect. In recent years, chitosan has been proven effective in preserving the quality of various food products. So it can be used as green and natural food ingredients. In this topic, I will focus on the findings related to the application of chitosan on seafood products and covering the factor influence specific bioactive property of the chitosan and application methods on chitosan-based active packaging to control foodborne pathogen in seafood products. Source of chitosan. The chitosan is a natural cationic polysaccharide. It is derived by the acetylation of the chitin. The chitin is mainly extracted from the crustacean, the exoskeleton of the crustacean, such as shrimp, the shellfish, the crab, and the mollusks, such as the squid pen, and the fungi and insects. But however, the main sources of commercial product for the chitosan are byproduct of the seafood processing. especially the crab and shrimp shell. Chitosan has a number of beneficial properties, such as being it's non-toxic, it's biodegradable, it's biocompatible, it's immunological neutral, it has antimicrobial activity, anti-inflammatory, and also some research prove that it has the anti-tumor with wound healing activity. So it has been applied in many industries, for example, in the biomedical devices, pharmaceutical, cosmetic, food, and etc. The chitosan and its derivatives are known to have a wide range of biological action. But for the functional property of the chitosan for food, mostly are focused on antioxidant activity, antimicrobial activity, film-forming property, and it can be used as the processing aids as well. In this topic, I will focus on the antimicrobial activity and film-forming property. We combine the two functional property. We can create the chitosan-based active packaging. For the antimicrobial activity of the chitosan, this is my research. I try to investigate the antimicrobial activity of the chitosan from different sources, from the shrimp, crab, squid, and comparing to different molecular weight, different degree of the saturation, and we found that, and I found that chitosan has a broad spectrum of the antimicrobial activity against most foodborne pathogens. This is some part, but actually I have tasted more than this. It shows the antimicrobial against food spoilage microorganism. It has the antifungal activity, but however, the antimicrobial activity of the chitosan quite varies, and it is specific to type of microorganisms. And furthermore, if we are considering only the chitosan itself, the antimicrobial activity depends on source of origin, either from shrimp, crab, squid, or mushroom. It depends on the molecular size, molecular weight, and the degree of the acetylation. But in normally, the higher of the degree of the acetylation, it's to show the stronger of the antibacterial activity. Furthermore, our lab has investigated the mode of action of the crustacean chitosan. We have studied the mode of action of the crab chitosan by using the HATR, or horizontal attenuated total reflectance spectroscopy, to monitor the reaction of the chitosan and the bacterial in real time. And we found that the first target organelle was the bacterial cell wall and the cell membrane. The interaction starts within 45 seconds. And after that, after the cell wall and cell membrane, then it's followed by the reaction in the proteinaceous and the DNA region. From this picture, you will see that the chitosan are stacked outside the bacterial cell wall, and for the next picture is more clear. And after treated with a higher concentration of the chitosan, it showed that the cell wall of the bacterial was leaked and the cell wall was damaged. there's many hypotheses on the mode of action of the chitosan, and this is the good example of the conclusion, the hypothesis of the mode of action on the antibacterial activity of the chitosan. The mechanism of chitosan on after the chitosan dissolved in the acidic solution, the chitosan molecule protonate to and it presents the positive charge of the chitosan molecule. Then it it has the electrostatic interact to the negative charge of the bacterial cell wall. For the gram-positive bacteria, it has the negative charge of the lipoteichoic acid on the peptidoglycan, and for the gram-negative bacteria, it has the negative charge of the lipopolysaccharide of the outer membrane. When the chitosan interact to the cell wall here, it can cause the collapse of the cell wall and cell membrane here, and then the chitosan can penetrate into the cell membrane, into the cytoplasmic, and then it can cause the proteinaceous damage of the protein and damage the DNA inside the cell. There are some hypotheses that prove that it has a hypothesis that the chitosan can inhibit the mRNA synthesis, the protein transcription, the protein synthesis, and also inhibit the enzyme. In some hypotheses show that the chitosan will has the polymeric envelope cover of the cell wall, so it prevents the nutrient to penetrate inside the cells. So the cell will lack of the nutrient and exhausted and die finally. With the film forming ability, the chitosan has the film forming ability, so it has been used as the food packaging. Incorporated with the antimicrobial activity, it can use as an antimicrobial film here, or when it forms the film, it can apply as the barrier film, or with in some cases with the more developed of the film, and it can be used as the sensing film. Most of them are most of the chitosan film, especially on the research, is a focus on antibacterial food packaging film. For the chitosan-base antimicrobial film, it has the antimicrobial film action by releasing the antimicrobial agent on the food surface. So it's widely applied in food. It can be applied in the meat, in the fruits and vegetables, in seafood. And I will show you some example on the application of the antimicrobial film in seafood and seafood product. The example, for example, in the ready-to-eat shrimp, the fillet salmon, and smoked salmon. These are research. This is the research on applying the chitosan-base anti-listeria film for ready-to-eat shrimp. It started with the cooked shrimp were artificially inoculated with the Listeria monocytogenes Scott A to demonstrate the post-process contamination. After that, the inoculated shrimps were dipped in the different chitosan solution, control that use for the control. There's uncoated with the film and coated with 1% acetic acid. These are the control samples. For the experiment, the chitosan solution, the concentration of the chitosan was 0.07, 0.07, 0.7, and 1% of the chitosan. After that, after dipped, the sample were let dry in the laminar flow cabinet, in the biosafety cabinet. After that, the sample were packed and stored at the 4 degrees Celsius. And this is the picture of the shrimp, ready to eat shrimp after dipped in the chitosan solution. And this is the result after store the shrimp at 4 degrees Celsius. The sample will withdraw every day and taste for the survival of Listeria monostogenus on the sample. The Listeria numbers were enumerated by overlay technique. The results showed that these are the control, both uncoated and coated with 1% acetic acid. The Listeria, these sample could not inhibit the Listeria growth, but when coated shrimp with the chitosan at the 0.07 and 0.7% of the chitosan can control the Listeria growth. It shows the bacterial static throughout the storage. And when increase the concentration of chitosan to 1% of the have increased the concentration of chitosan to 1%, it can reduce the number of the Listeria contaminate on the shrimp throughout 10 days. And this is an example of the chitosan-base antimicrobial film applied for the salmon fillet. This is the research paper that I show you. The salmon fillet was coated with chitosan at 1%, and the fillet was inoculated with many with the coliform, aeromonads, vibrio, and pseudomonads. For the aeromonads and vibrio, it's related to the foodborne pathogens, and pseudomonads and coliform, it is the spoilage microorganisms. The results show that the chitosan film can reduce the vibrio growth, the aeromonads, coliform, and it can inhibit the pseudomonads, the growth of pseudomonads for a couple of days. After that, the pseudomonads can grow quickly. This is another example of the chitosan film, chitosan coated on the plastic film, incorporated with the additive, the GRS, generally recognized as safe additive, with the combination with the chitosan film and the food additive. It can increase, it can enhance the antibacterial activity of the it's better than using the chitosan itself. This shows the inhibitory effect against Listeria monocytogenes on cold smoked salmon that store at 4 degrees Celsius. For the plain film and chitosan film show less effective against the Listeria in this case. But when the researcher added they incorporate with the other additive. In this case, they add sodium lactate and potassium sorbate. You will see that when adding some additive into the chitosan coated on the plastic film, it showed a better result, and it controlled the Listeria growth for over 8 days. Furthermore, the chitosan can be applied. Chitosan can combine with other biomaterial to develop sensing film, which with the sensing film sensitive to pH, microbial enzyme, and microbial metabolism. In this picture, it shows the colorimetric sensor, which is popular currently. This research, they incorporate the chitosan with the anthocyanin. The anthocyanin is very sensitive to the pH. So the color of the anthocyanin will change if the pH of the sample change. The anthocyanin is immobilized to the modified film for indicate the pH of the food. In this research, the chitosan and anthocyanin-based colorimetric pH sensing film was developed, and it showed the color from red to green. It reflects to the freshness of the fish. In another,
Dr. Chitsiri.
Hello.
5 minutes left. Yeah.
Oh, okay. Thank you. It's almost finished. This is another example of the microbial sensor. The chitosan hydrogel film with a functional of the beta-glucuronidase secrete by E. coli strain. It used the same principle to the selective medium. The researcher add MUG, which is the methyl-lumberiferyl beta-D-glucuronide into the hydrogel film. And when the E. coli grows and it secretes the enzyme of beta-D-glucuronidase, so it breaks down the substrate, and it can change the color of the film from colorless to yellow. If the film changed to yellow color, it shows that there is the E. coli contaminant in the product. In conclusion, I have shown that the chitosan has lots of functional property for food, and it is widely used in the food industry. The chitosan-based film can be applied as active and intelligent food packaging for seafood safety and also for the food quality. Thank you for your attention.
Thank you very much, Dr. Chitsiri. How to say in Thai? Khop kun kap. Thank you very much. So, all audience, we already hear 3 great women researchers today. So we will go to the question and answer. But before that, yeah, we will have a photo session first while waiting for you to looking for a question. So for those who are able to open your camera, please open the camera, and we are requested the IT team to take the picture. IT team, please.
Okay.
Yeah, thank you. So please, for those who are able to open the camera, please open the camera.
Okay, ladies and gentlemen, I will take a screen in count 1, 2, 3. Okay, 1, 2, 3. Thank you.
Thank you very much. So for those who have question, we have 3 researchers here. First speaker is Dr. Indun, talking about the histamine contamination and how the best way to prevent histamine contamination. And the second speaker, Dr. Lotta, talk about an interesting method and an advanced method on how we're detecting fish spoilage by using the volatile organic compound. And our third speaker, Dr. Chitsiri, talking about Chitosan and the application on a packaging material. So you can raise your hand or you can write your question in the chat column. We are waiting for your participation, please. For Indonesian participation, if you want to speak in Indonesian, it's also welcome. I will translate to English. Okay, while waiting for a question from the audience, then I will ask a question for Dr. Lotta. It's interesting to talk about the volatile organic compound. Could you explain more about how if the sample is not fish, maybe it's shrimp or oysters, is this the same characteristic or will be different characteristic?
Yes, it's a very good question indeed, because the type of the product will definitely have a great influence on what kind of spoilage processes there are. So on the VOCs as well. So in our research group, we have done research with different kinds of seafood products. So indeed, also including shrimp and mussels, for example. So there are many similarities. So in products where microbial growth and chemical reactions are the prominent reasons of spoilage, the principles of measurements are often similar. So we can use similar kinds of methodologies for analyzing the different types of products. But indeed, the VOCs which emerge, they are partly the same often. So like, for example, indeed, ethanol is a compound which we very frequently detect under different conditions. But on the other hand, then there are some compounds which are very specific to a certain product type. So overall, I would say that the methodologies generally can be applied for many kinds of products, but indeed what we get as a result, that can be partly similar, but partly different. So that's why it's always so interesting.
Okay, thank you, Dr. Lotta. Okay, for those who have any question, please, you can write a chat in the chat room or just simply raise your hand. Okay, we have a chat. I have a chat from Bastian. Bastian will want to ask to Dr. Chitsiri. So okay. So he wants to know about the chitosan that is dissolved in acidic. So is it affect the taste of the food? Thank you.
When we use the chitosan, we have to dissolve in the acidic acid. In the acidic condition, we can dissolve in the lactic acid or acetic acid, but it's dissolved very well in the acetic acid. And I have proof that if we use less than 0.3% of the acetic acid, it have no effect on the sensory evaluation based on the starch food and on the starchy food, for example, the noodle And for the seafood, yesterday I just did the research. We do the sensory. We add 1% of the acetic acid to coat it on the salmon fillet. It's no effect at all. Yeah,
Okay.
the evaluator cannot detect it.
Okay, thank you very much, Dr. Chitsiri. Again, for those who have a question, you can raise your hand. Okay, another direct message to me from Atar. Atar want to ask to Dr. Indun regarding Indonesian as a tropical country. Is the tropical temperature affect the histamine? Please, Dr. Indun.
Okay, thank you, Mr. Atta.
Yeah.
Okay, Mr. Atta, thank you very much for your question. Actually, the main one of the main factor which affect the production of histamine in fish is the temperature. So that is the big challenge for the tropical country to maintain the temperature in a low temperature, or we can say a cold chain during all the step of the handling and the processing for fish. But actually, in fact, when we saw in the Sadeng fishing port, and also I believe in many fishing port which is used, or the technique which is used by the small fishermen, I think cold chain is still difficult thing to be practiced. Because usually they use ice when they go for the trip, but sometimes ice is already melted during the trip, because some ship they use, or they have to go like 5 or 1 week for trip, and some they use salt for maintaining the temperature of ice. But in fact, after the fish came to the port, and then they put out all the fish, the step of, or the handling step after take out the fish from the ship is not maintained for the temperature. And I think that is one of the chance for the bacteria to produce the histamine during that time, when we see that the time they use, or the time they try to handle all the fish until they get back the fish into the ice is around 200 minutes. I think it's quite a long time. And when we try to do, but this is in the laboratorium condition, in like less than 24 hours, for the 30 degrees and sometimes 20 degrees, the bacteria can already produce the histamine. So I think the key of the handling is the temperature. So that is, you are correct, the big challenge for the tropical countries.
Thank you,
that is my
Dr.
answer.
Okay. Okay. So, okay, another chat from Susana Endah. Yeah. Thank you for the presentation. I enjoyed it very much, Dr. Chitsiri. I saw colorimetric test for fish product, which show the color sticker change from red to green. Yeah. Could it be applied for another seafood? Yeah.
I think it has potential to be used in other foods if it have the same of the pattern. If it had the same pattern of the spoilage, for example, it is spoiled because of the pH. It can apply it. And anyway, it needs more further adjustment, or it needs the validation as well. Yes.
Okay, thank you very much. So it's still wide open to the application, for the application. Thank you, Dr. Chitsiri. Okay, we still have 44 participants here. Is there any question or comment? Comment is also welcome. Yeah. Yeah, we still have from Myanmar. Can you say hello? From Myanmar. Good afternoon.
Good
Okay.
afternoon.
Okay, we still have from Malaysia. Malaysian participant, maybe you can say hello. Again, if you have a question, you can raise your hand or direct chat to me.
May I have question?
Okay. Please, Dr. Indun.
Because I'm also participant. I would like to ask to Dr. Chitsiri. I saw your presentation about the type of chitosan from the different sources of organism like fungi and also crustacean, and they have the different type of chitin or chitosan. What is the factor of this different type or different structure to the antimicrobial activity? Is it can give any effect to the antimicrobial activity? Thank you.
I found that the pattern of the chitosan affect to the antimicrobial activity. For example, for the squid pen, the pattern of the chitosan is beta. It's for the other chitosan source like the shrimp and crab, it's alpha. The antimicrobial activity is different, and I found that for the Bacillus cereus, it's quite sensitive to the crab chitosan more than it's sensitive to the crab chitosan than the shrimp chitosan. But I don't know what. I don't know the reason yet. I know just the result. And I found that comparing from the crustacean chitosan and compared to the mushroom chitosan from different source has the strength of the antibacterial is totally different. And I found that for the Thai mushroom has the stronger antibacterial than Chinese mushroom. And I try to compare by looking at by using the FTIR, looking some pattern of the biomolecular, but I saw some structure of the mushroom chitosans are quite different, but I don't know exactly how and what. It need more further investigate. A lot of things to do.
Okay, interesting. Thank you, Dr. Chitsiri. Okay, audience, we still have 5 minutes left for the question and answer. Again, if you have a question, please raise your hand or write on the chat account. Okay, maybe another question from me to Dr. Lotta. It is interesting to hear about the gas detection. Is it possible if we make like a prototype for detecting the gases? Thank you.
A prototype for detecting the gases. Yeah. So
like a kit, yeah.
What kind of prototype?
Like a kit, a machine or something that we can bring to the market, and we can test the fish freshness.
That's indeed the principle behind, for example, the building of sensors and indicators of them, because indeed in laboratory scale the measurements have been possible for already quite some time, and there are many methods, but a challenge is how to then convert this technology, for example, to a sensor which could be used not only in the laboratories, but also, for example, during the distribution chain. So thinking of the distribution and for example, supermarkets. So this is indeed the challenge there is that we need to indeed collect data at laboratory conditions and then transfer this data so that we can indeed build sensors which are able to achieve the monitoring. So indeed, this is this is a very hot topic, has been for quite some time already, and there are many interesting applications already and studies which have arisen from this. So indeed, looking very interesting.
Big challenge. Thank you.
Yes, that's not true.
Yeah. So unfortunately, we are running out of time. So please give a big applause to our three speakers today. Thank you very much for coming and for sharing your latest research result to our participant. So that's the end of today's session. On behalf of the committee, I want to say again, thank you very much for Dr. Indun, Dr. Chitsiri, and Dr. Lotta for coming and sharing your knowledge to us. And after this, we have 15-minute break. And after that, we will have a discussion, all participants with our instructor today, Dr. Siti Aribudianti and Dr. Annie. And I'm as a moderator, say sorry if I have make a mistake. Thank you very much. Good morning to Dr. Lotta and good afternoon for all of us. Thank you.
Thank you very much.
Thank you.
Thank you.
And we have to see you again in the next,
Yeah.
or maybe not online, but offline.
Line.
Yeah. You are very welcome to come to Yogyakarta. Thank you.
You are very welcome to Kasetsart University too.
Okay, thank you very
Looking
much.
forward to see you both directly someday.
Yeah.
Thank
Bye.
you very much. Bye-bye.
Okay,
Bye.
please, for all participants, please come again and be active at 4:05. Thank you.
telepon. Hello, Sita. Can you hear my voice?
Yes, clear.
Okay.
Okay, we can start now, the committee. Yes, we start now, Bu Eni.
Okay.
Okay, yes, I will start now. Good afternoon for all participants this afternoon. We still have 28 participants. Okay, I will introduce myself before. My name is Siti Aribudianti, or you can call me Sita. This afternoon in Indonesia, I will accompany you, all of you, with Dr. Eni, and we talk about the Spirulina platensis as kind of cyanobacteria that can fortify the fisheries product. Before we start the discussion, the committee will play the video. It can stop now, but Desi. After that, we have a Kahoot activity that will conduct by Dr. Eni. We can start now. I hope all participants will enjoy the video. Thank you very much.
Give me a minute.
Yes, yeah, Mas Yafi. Are there a problem?
Spirulina atau Arthospira sp merupakan salah satu kekayaan alam yang dimiliki Indonesia dan banyak tersebar di berbagai wilayah perairan Indonesia. Spirulina dikenal sebagai superfood karena kandungan nutrisinya yang lengkap seperti protein, vitamin, mineral, dan antioksidan yang bermanfaat bagi kesehatan. Selain itu, spirulina juga dikenal sebagai sustainable diet karena dalam siklus pertumbuhannya, ia dapat menyerap emisi karbon dioksida dengan efisiensi yang tinggi, tidak membutuhkan banyak air, serta mampu tumbuh pada lahan marginal. Sehingga, budidaya ganggang hijau biru dari kelas cyanobacteria ini dapat mendukung kelestarian lingkungan. Sebagai bahan pangan kaya nutrisi, spirulina mampu tumbuh sangat cepat dengan efisiensi produksi yang tinggi. Universitas Gadjah Mada ikut mengambil peran dalam pengembangan mikroalga spirulina untuk aplikasi di berbagai bidang. Melalui Pusat Studi Energi bersama Nogotirto Algae Park, UGM telah mengembangkan fasilitas kultivasi mikroalga spirulina dengan kapasitas penuh 100.000 liter di Yogyakarta. Berbagai produk telah dikembangkan oleh Nogotirto Algae Park seperti biocrude oil, sabun, masker, hingga suplemen. Selain itu, kandungan spirulina yang kaya akan nutrisi dan senyawa bioaktif yang bermanfaat menjadi dasar pengembangan spirulina sebagai pangan fungsional berbasis hasil perikanan unggulan saat ini di Universitas Gadjah Mada. Salah satu tantangan yang dihadapi dalam pengembangan produk pangan fungsional berbasis spirulina adalah terkait stabilitas senyawa bioaktif selama pengolahan dan penyimpanan, serta adanya rasa dan aroma khas yang dapat mempengaruhi penerimaan konsumen terhadap produk. Laboratorium Teknologi Pengolahan Ikan Departemen Perikanan Fakultas Pertanian Universitas Gadjah Mada telah mengembangkan berbagai penelitian berbasis spirulina yang dapat diterima dengan baik oleh konsumen dengan tetap mempertahankan kandungan gizi dan senyawa bioaktif yang berkhasiat. Mahasiswa Teknologi Hasil Perikanan telah mengembangkan minuman coklat spirulina di Laboratorium Teknologi Pengolahan Ikan Departemen Perikanan Fakultas Pertanian Universitas Gadjah Mada. Minuman coklat menggunakan bahan-bahan berupa bubuk Artospira platensis, bubuk kakao, krimer bubuk, vanili, tepung rumput laut, garam, dan gula pasir. Selain pengembangan di skala laboratorium, spirulina juga dapat difortifikasi ke dalam berbagai produk pangan olahan masyarakat. Salah satunya adalah bakpia spirulina. Bakpia Mino Istu menjadi salah satu produsen bakpia yang menggunakan spirulina dalam pengolahannya. Bakpia sendiri merupakan makanan khas daerah istimewa Yogyakarta yang autentik, unik, dan ikonik. Bakpia merupakan makanan klasik tradisional di Yogyakarta yang merupakan wujud nyata akulturasi budaya Cina dan Jawa yang telah berlangsung ratusan tahun. Nilai sejarah dan budaya ini merupakan sebuah kekayaan dan bukti kelangsungan toleransi di Yogyakarta dan Indonesia. Seiring dengan perkembangan zaman, produk bakpia telah mengalami berbagai inovasi yang pada mulanya bakpia berisi kacang hijau. Saat ini berbagai rasa kekinian dapat dijumpai dalam bakpia seperti coklat, keju, green tea, red velvet, dan lain sebagainya. Pengayaan kandungan dan nutrisi spirulina yang dikombinasikan dalam bahan-bahan lokal untuk olahan bakpia menjadi inovasi baru dalam pengembangan produk pangan khas daerah Yogyakarta dengan basis hasil perikanan.
Okay, thank you for the committee. I think it's very interesting video. I hope all participants enjoy the video. After that, Dr. Annie will continue with the Kahoot activity for all participants. I think there is still 32 participants. I hope we all enjoy the Kahoot activity for this afternoon. Dr. Annie, will you start now?
Sit down for the time.
Yes,
okay. I think it's good because the participant increase in the number. So, hello everyone. After we watch the video, let's have fun for a while with a little game. I will share Kahoot and we will play with Kahoot. Please prepare your device, handphone, computer, or laptop that we will use with the Kahoot game. I will share my screen. Can you see my screen, Puspita?
Yes, clear.
Okay, please every participant, please enter the Kahoot with www.kahoot.id and use this code to enroll in this game. Or also you can scan this code to enter this game. Okay, we will give you time to enter this game. Okay, Pak Edi Trihatmoko has joined with us. Okay, the other participant for committee also can join with us in this game. Just let have fun. Puspita, maybe you can also join in this game.
Yes, I have joined, Mbak Eni.
Okay. There is Awa. Who is he or he?
Maybe
Okay.
one of the problem is connection, Bu Eni. Maybe we can wait around 1 minute more.
Okay.
Yeah, okay.
Hi, participant. Please join. Okay. During writing, I will explain how to play this game. Every participant, please answer every question in this Kahoot session. We have 6 questions. There is several types of questions, multiple question, and also fill the blank question. So please be focused, and we will have start this game soon. Okay, we have 14 participants, 15.
Yeah, let's join. All of the participants is still 32.
Okay. If you have any problem, let us know your problem. I think it's quite easy to join with the Kahoot. Okay. We make like some competition, little competition. Every right, correct answer and the fast answer will get the highest score. Okay, maybe 2 minutes. We will wait you.
Yes, okay. For 30, I think, Bu Eni, we can start the game.
Yeah, okay.
Yes.
If you feel shy, you can use your alias name.
Okay, now 19 participants. Thirty seconds, Bu Eni.
Okay. Okay, 20. Don't worry, all question is we take from the video. So I think it's quite easy.
Okay, now 21.
Okay, 1 minute more.
Could we start now, Bu Eni?
Okay. Okay, please
Yes.
be focused and enjoy this game and good luck. I will start the game. Okay, the first question. Spirulina belongs to the group of, you have time 30 seconds. Okay, time is up. There is 18 correct answer and 5 false answer. So the correct answer is cyanobacteria. Okay, we will know who the high rank. Okay. Oh, Sop. Who is Sop? Sop is get the highest score. So for the next question, are you ready?
Yes, ready, Bu Eni.
Okay, for the second double point. Spirulina is also called blah, 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 blah. Please type your answer. Minute left. 5 seconds left. Okay, 40. So there are, oh, okay. There are options and there is no one that can type the right answer. Spirulina, the other name is Artospira platensis, Artospira, or you can sort with A platensis. Okay, so I think the score is still same, so still the highest. Okay, we move to the next question. Okay, the third question, double point again. What is the meaning spirulina as superfood? Ready to answer? Okay, time's up. Okay, all participants, great job. All participants can answer the right. Okay, who in the first? Oh, sorry, Sobrina. Yeah. Okay, Chogi has get the first rank. Okay, the other participant, let's we have fun to get the highest score. We move to the next question. Okay, let's go. The fourth question. The following statement is related to sustainable diet, except okay. Who will in the first rank? Okay, there is 12 participants that can answer with the right answer. So who will be the first? Oh, IT Fisheries. Okay. So the other, let's overtake the score. Okay. The next question. Are you ready? Okay, next. 5. Question. The challenge on food product except exception. Okay. Oh, maybe you track with this question. There is except word. So I think all participants still answer with the right answer. So it's false. The truth is hard to cultivate because except there is except word. Okay. Who will be the first podium? Oh, Mascobis. Who is Mascobis? Okay, good job, Mascobis. And the next, maybe the last question,
Yes, as
Okay,
the last
for-
question, Benny.
Yeah, okay, we will play. Okay, this is the brainstorm question. An idea about the product that can be developed from Spirulatensis. Every participant can type up to 3 ideas, product idea. You can type the product that decided explained in the video. Okay, the time we give to rather longer than the previous question. Okay, let me see. What has answered the question? Sorry. Second left. Okay, we open the result. Okay, 1, 2, 3. Okay. Oh, there is bakpia, cakes, bakpia, dim sum, cookies, bread, spiral cakes, chocolate drink, biscuit, crackers, pudding, powder. So many products that you can see in this screen. So please read carefully all the products because we will move to the next question. One more bonus question. Okay, pizza drink, face mask, packaging material, vehicular fuel, fabric and textile, medicine, yogurt, bread, chlorella. Okay, for the next, please read carefully all the product and let's start voting the product that the most you want to add with spirulina. I will click the start voting and you can choose which one that you most wanted. Okay, we start 1, 2, 3. Okay. Vote to the best according to you that will give the pleasure taste to when it ate with the spirulina. Okay. Still time to put 1 minute to vote. Okay, time's up. What the product that most you want? Okay, we refill it. Okay, ice cream. Okay. Oh, and also drink, ice cream, drink, and bakpia and cookies. Oh, the winner is cookies. Okay. Okay. And for the last Kahoot, we will reveal who will the winner for this session. Okay, the podium. We show. Okay, the second Johannes Alpin, and the third Kitchen Era, and the first Maccabees. Great job. Okay. Okay, this is the Kahoot session. So, Puspita, you can continue the discussion.
Okay, thank you, Benny. We still have around 18 minutes. Okay, so the winner is Mascobis. Can I know who is Mascobis here? Okay. Yes, okay. From the discussion, the favorite product for spirulina fortification is bakpia, ice cream, and cookies, I think, Bu Eni. Maybe we can discuss. Maybe I will ask Mas Johannes here. Mas Johannes still here? Mas Johannes, do you choose ice cream? I think at the spirulina fortification here. Hello, Mas Johannes. Okay, there are no answer. No response. Maybe I will ask Mbak Desi here. I think Mbak Desi choose cookies, Mbak Desi, for kind of product that will fortified with spirulina. Okay, Mbak Desi. Why? Yes. Why do you choose cookies here for spirulina fortification, Mbak Desi?
Yeah, because actually I don't like as much as chocolate, so maybe it is better for us to kill some another flavor like spirulina for the cookies. Yeah, I think the flavor is more wonderful.
Okay, thank you, Mbak Desi. Yes.
Thank you.
How about the bakpia? Maybe there is a participant that choose bakpia as a product that well fortified with spirulina platensis. Are there any participants will explain?
Maybe Mas Eddie?
Okay, maybe Mas Eddie. Mas Eddie is still here?
Yes, Puspita, I'm here.
Okay, how about Mas Eddie? What kind of food will you fortify with spirulina platensis, Mas Eddie?
I do remember that I wrote bakpia for my option. Yeah.
Okay, yes. Oh, why do you choose bakpia, Mas Eddie?
Originally I came from Jogja and I love eating bakpia and I love to share bakpia to my colleagues from other cities and it's quite good.
Okay, yes. Thank you very much, Mas Eddie.
My pleasure.
Yeah, thank you very much. So there are two products that has explained by Mbak Desi and Mas Eddie. They choose bakpia and cookies. Mas Johannes, are you still here? Why do you choose ice cream, Mas Johannes? Okay, there are no answer from Mas Johannes. Oh, okay. We will continue now about the fortification product or product that fortified with spirulina. Bu Eni, let's continue the discussion for this afternoon. We still have 15 minutes.
Okay. For participants, I want to know, have you tried the product that fortified with spirulina? Please raise hand, the one that ever tried the product that fortified with spirulina. Oh, there is no one. Oh, okay, Mas Eddie. The other? Okay.
Also, yes, Mas Edi.
Okay, Mas Edi again. Mas Edi, can you share with us how the taste and the flavor the product that fortified with the spirulina?
Actually, it was my mom. So my mom bought a spirulina face mask, and then the smell is so bad. But my mom said that it's so good for our face, so that's why I tried it. Then maybe it has a really good advantage for us. So yeah, I will pursue that again.
Okay, so you can more detail explain about the flavor is bad.
The smell,
What's look like?
the smell is bad.
Okay, the smell bad. What's look like?
Uh, I don't know, but I think that I have a good smell. I mean, I have a good nose, and it's really bad. I don't know how to explain that. Bu Henny.
Okay.
Is it like a fishy flavor, Mas Edi?
Like a what? Sorry?
Fishy flavor. What
Oh,
is
fishy
it like?
flavor.
Yeah.
Yeah, it's a little bit fishy flavor, and I can smell the ocean from that. And I can smell the grass pregnant. And I also can smell the, how to say, like, yeah, a rotten fish. Sorry for saying that.
That's right. That the spirulina has the flavor or aroma that like fishy. And also maybe the other ever tried to taste the product that fortified the spirulina? No one? Okay. In the previous video, there is statement that spirulina has distinctive aroma, flavor, and taste. So spirulina basically has the bittern bittern taste because it has if we add the high concentration of spirulina, it will cause it more uncomfortable for the consumer. So if you have the way to overcome this problem? Can you imagine? There is the taste, bitter taste, and also fishy flavor. Do you have idea how to overcome this problem? For example, if you add the bakpia with the spirulina, can you imagine how to overcome in order we get the product bakpia that not too bitter and also fishy? Hello, maybe the other participant. I think Nur Asarmin from Bangladesh. Hello. Okay, hello. Hello. I think the first participant that get the highest rank. Sobrina Silmi. Sobrina, are you there? Oh, there is no one responding. Okay, maybe the other participant. Okay, maybe Puspita, you will answer this question.
Okay, I will answer the question from Dr. Annie. I think there are many kind of product that can mask the fishy and a bitter taste. Maybe for bakpia, we can add the vanilla flavor to decrease the fishy taste. And also we can add like cinnamon if we add Spirapatensis as cookies fortified that the cinnamon that we add to the cookies or ice cream can decrease the fishy flavor at the product.
And the second one, for decrease the bitter taste, sometimes the activity at our laboratory also add stevia to decrease the bitter taste of the product that fortified with Spirulina platensis. I think it can answer the Dr. Annie question for the how to decrease the fishy flavor and how to decrease the bitter taste of the Spirulina platensis.
Okay.
How about that,
Thank
Bu
you,
Annie? Is clear? The question, the answer is clear?
Yeah, very clear, I think.
Yeah, okay. I hope all participants clear and understand about that. Okay. So we continue. We still have 8 minutes here. I will continue about the idea of the unique product from the country for its participants. We still have from Malaysia, and also here we still have participants from Bangladesh and Bahama. Maybe I will call Mr. Shafiq Aiman from Malaysia. Mr. Shafiq? Oh, no answer. Another participants from Bangladesh, maybe? Miss Nur Asharmin, could you answer for the kind of product that is favorite at your country and it could fortify with Spirulina platensis? Or maybe Mas Harya Bimasuci from Indonesia? Do you have an idea to another product that fortified with Spirulina platensis? Mas Harya is still here?
Oh, yeah.
Yeah, okay, thank you, Mas Harya. Do you have an idea? What is it, like a unique product from Indonesia that can fortify with Spirulina platensis?
Last time I ate something with spirulina was, I think my friend Nita, she just baked some cookies with spirulina. It was quite delicious, but when she added like chocolate flavor, it is not very, I don't know, it is not very chocolatey, but the coloring is very good. And back then, when I was in Undip in my S1 days, one of my docent kind of produced an ice cream from spirulina. It has vanilla flavor, and also the flavor was enhanced with cinnamon, and the coloring is very great because of the phycocyanin.
Okay.
It is, I think it is equivalent to brilliant blue coloring, but the smell is the cinnamon cannot mitigate the smell.
Okay.
I think we should, I don't know, we should innovate something to mask the smell, and also the fishy taste is. I think the taste is quite significant even with vanilla flavoring, but I don't know, maybe we can apply the spirulina in like fruit ice, or maybe something ice with some fruity flavoring to mask the flavor. I think, yeah, fruity ice with spirulina. I think that should be good.
Okay, thank you very much, Mas Harya. Maybe there is a good idea from Mas Harya to masking the flavor. At our laboratory, we also has like nano capsules from carotenoid from Spirulina platensis that after we get the nano capsule product, then we add the nano capsule to the product like ice cream and also cookies, and it can decrease the fishy flavor, and also it can increase the customer acceptance. So our laboratory tried to use nano capsule product to fortify the, what is it, like many nutrients from Spina platensis at the product with nanocapsules. I think it's from the idea from our laboratory. We still have 4 minutes, Bu Eni. Maybe there are questions, still a question or discussion this afternoon. Thank you, Mas Haria.
Okay, maybe the last question from me. Mas Haria said that the spirulina gives the blue coloring. Is there any participant that know why spirulina has blue coloring? What is the substance of spirulina that causing the spirulina has the blue coloring, blue color? Is there any participant know about this? Okay. I think who from hello, Mbak Susan. Still there? Are you still there? I think it's not joined. Okay. Okay. Maybe for the time I will share. But why the spirulina can be the color, give the. Oh, okay, okay. Miss Susan answer because the blue green algae. What is Puspita? Okay, good answer, Miss Susan, Miss Mbak Susan. Because yes, spirulina is blue green algae. Why it called blue green algae? Because the spirulina contain with the phycocyanin. Phycocyanin is blue with color, the color is blue, so it can be used as the blue coloring in the food, and it also can be used as antioxidant. Okay, I think that's the more information from me, Puspita.
Okay, thank you, Bu Eni. I think the time is up. There are still 2 minutes. Today we will have discussed about all of the, what is it, like nutrition, composition, and superfood of Spina platensis that can fortify at any products that we hope the products that we eat will, what is it, like increase our healthy. Okay. Okay, for today, I think it's finished. Thank you very much for all of the participants that have joined from 2 p.m. at Indonesia. We hope all of the participants will join again tomorrow at 2 p.m. Indonesia, and maybe around 3 p.m. at Malaysia, and I think at Myanmar too. For tomorrow, we will meet again. On behalf of the committee, thank you very much for the participants. And I'm sorry for all mistakes that me and Bu Eni do this afternoon. Thank you very much. Let's meet tomorrow for the summer course at Universitas Gadjah Mada.